Welcome to the Toxicology Lab at Valley City State University. My name is Hilde van Gijssel and I'm a professor at science here at Valley City State, and I'm guiding you through a tour of our research lab. This is the lab that our students use to do research, and there's pretty much two parts to it, and I'm going to walk you through the first part. On this side of the lab we are doing our biomedical stuff. So this is where we work with our cells, we do our stainings. We isolate DNA, proteins, and RNA. As you can see, of course, every lab has to have a sink so that we can clean our stuff, so that we don't get contamination. This is our workbench. This is where we actually do the work. And then on this side, we have pieces of equipment that are important for our experiments. So we have a plate reader. This is a machine where we can look at ultraviolet light and see DNA in gels. You can see we have all of our glassware. We have heating blocks, cabinets to mix things and keep them on temperature. This is our PCR machine. This is a new tool that was developed in the 1980s and changed science forever. This is where we can multiply DNA in a test tube, and we can make more DNA when we need to. This is a machine that can count cells in a solution. So if we need to have a specific number of cells, we can determine that and then know how many cells we plate, and then we can test how many die if we expose them to chemicals or how they survive. Of course, we need to have balances and hoods for things that are dangerous. And then I'm walking you guys to the other half of the lab, and this is what we call our fruit fly lab, or Drosophila lab. We do experiments with fruit flies, and fruit flies are little flies, and if you want to manipulate them, you have to put them under anesthesia. And interestingly enough, we use carbon dioxide for that. And so these are the little beds that we use, and they fall asleep in about 30 seconds, and then you can look at them, determine their sex, traits that they have, and we can use microscopes to do details. And in class we do an actual experiment with them where we look at the Mendelian genetic patterns of inheritance. And fruit flies are really cool to do that. The other thing that we do with the fruit flies is that we look at multi-generational exposure, and with this setup we are able to do that. And on this side you see that this is nice and clean because right now we're not having fruit flies, but this is where we can make the media and their food that they live on, things like that. And then in this corner we have our spectrophotometers. We have an equipment that can measure bioluminescence, and then of course the traditional chemical cabinet, our refrigerator and our freezer. These are all the important tools of biomedical research. They don't look very interesting on camera, but if we take them, we can generate beautiful little pictures and see what happens to cells when you treat them and expose them to all kinds of chemicals.